So we're talking about reproduction. We ended our notes by beginning to start talking about chromosomes, but as we start, we just want to get a little bit of a review. Where is this? What exactly are we talking about? Remember, we're talking about reproduction, which can occur either sexually or asexually. We're choosing to focus first on asexual reproduction and the advantages of asexual reproduction. Remember, asexual reproduction is usually used for three specific reasons. One is to reproduce. If you're a unicellular organism and you divide your cell, all of a sudden you've reproduced. Some multicellular organisms will use asexual to reproduce, and the advantage is you get a clone of yourself really quickly. We are going to talk more later about the advantages of sexual reproduction, but asexual reproduction is essential for multicellular organisms because it's how multicellular organisms grow and repair themselves. When we want an organism to grow or repair, we want those cells to be identical to the other cells of the body. And so that is the advantage of using asexual reproduction. Now, the cell division that uses asexual cell division is going to be mitosis. So we are going to get into reviewing mitosis and adding some new thoughts in. But there's something we need to know before we go on, and that's really understanding what this chromosome represents. And you see a chromosome, you know it's made of DNA. But there's a little something else that we want to add in, and it's how is it that DNA is wrapped up, right? It's not just smushed together, because that's what it looks like, just smushed. It turns out that the DNA is actually wrapped around a protein called a histone, Histone, a histone protein. And that histone protein needs to somehow attract the DNA. Now we know a little something about DNA. We know that DNA has the sugar phosphate backbone. And if we have phosphates that are negatively charged, this actually makes DNA negatively charged. So these proteins are actually positively charged in order to attract the negatively charged DNA. So when we see a chromosome and we see that it's coiled and packaged up, we really need to know the reasons why. One of the reasons why is because we're preparing for cell division. We need to be able to move our DNA in an organized way. And in order to do that, we use the chemical nature of the DNA molecule. So, when you look at a chromosome, you should know a couple of things. When you see a doubled chromosome, that will tell you that the cell is preparing for division. Interestingly enough, we talked about how our chromosomes come in pairs. These pairs are called homologous pairs. And we'll talk much more, that's G-O-U-S, we'll talk much more about homologous pairs later. One thing we do want to know for right now is that homologous pairs are found in sexually reproducing organisms. If you remember from meiosis, we're actually going to separate these when we make our sex cells. Homologous pairs, one of each. One came from mom and one came from dad. So we'll talk a little bit more later about these homologous pairs when we talk about sexual reproduction. But there is something that we need to know for now, which is this prefix homo means the same. Now, what of these is the same? Because not everything. Obviously, one is represented as purple and one is represented as green. They are usually about the same size. And they will have the same sequence of genes. For example, this might be the section of DNA that gives us eye color. And this might be the section of DNA that gives us color blindness. They may have different versions of the gene. And again, we'll get more into this later. But we might have a capital letter and a lowercase letter, if you remember alleles. What you need to know for right now is that when you see chromosomes, you should be able to make a prediction about the organism and about what's happening to that organism. So when I see these chromosomes, I see that they're coiled up, but I don't see that they're copied. If I see the double chromosome, I know that they've been copied and that they're preparing for cell division. And so that's one of our indicators that a cell has actually copied its chromosomes. We want to look a little bit closer at what the DNA might look like in different instances. For example, if we see a cell with DNA that looks like this, this is actually called euchromatin. And you might recognize the prefix "u" as meaning true. Chromatin is relaxed DNA, meaning not coiled and packaged and folded. And the advantage of relaxed DNA is you can access it for making proteins, right? We need the code for proteins. So a cell that has euchromatin is not preparing for division. This cell is going to be what we're going to call interphase, which you might remember. Of course, it turns out interphase is much more complex than you learned in Biology 1. But this cell is not dividing right now. However, it might receive a signal to divide. And that signal will start this process. When our DNA is coiled DNA, we call this heterochromatin. And we can begin to clearly see the doubled chromosomes. An interesting thing is you should be able to count the number of chromosomes. For instance, this cell, now that it's coiled, we can actually see that it has 1, 2, 3, 4 chromosomes. Now be careful. We do not count this as 1, 2, 3, 4, 5, 6, 7, 8. Because if you remember,
Briasia James, please come to the front office. Briasia James.
if you remember, this X shape is the doubled chromosome. And the two sides are just copies. So we don't count it as new DNA. So this cell has 4 chromosomes, 2 homologous pairs. You see 2 big and 2 little ones. And so we should be able to make some predictions about what might happen to this cell. Now, one quick thing is this doubling of chromosomes, we're not doubling the number, but we are doubling the amount of DNA. It's just that the 2 DNAs are exactly the same. So if they were asking me in terms of grams or in terms of an amount, I definitely would have double the amount of DNA. We just don't count it as new chromosomes since they're identical to each other. So you should be able to begin looking at pictures of cells and identify what happened before and what will happen after. But this all hinges on the fact that the cell needs to receive a signal to actually do this. And so we want to talk just a little bit about this signal. Pay attention to what this says. Normally functioning cells receive a signal to divide. Now this signal, we've actually talked about signals like this. If you remember FGF2, fibroblast growth factor 2. It's a signal that will bind to the outside of the cell and will actually tell the cell to begin to divide. So a normal cell doesn't do anything until it receives a signal. We're going to talk much more about this cycle, and we're going to talk about how these cells should also follow directions, right? When we talk about cancer cells, it turns out that there's some sort of breakdown in the communication. What we want to know for right now is that this whole called the cell cycle. Now you probably remember interphase and you probably remember mitosis, but there are a few important things that we need to know. G1 is the initial growth. So it turns out that interphase is divided into 3 sections. Interphase is the longest of the cell cycle. And that makes sense. You're going to do a lot of prep work, and then you're going to hurry up and divide. So the initial growth happens during G1. And then when we receive this signal, then the cell will actually synthesize or replicate more DNA. And so we call this the S phase. After we've synthesized the DNA, this is when we copy the rest of the cell, like the organelles. And then only if all of this has gone right will we go through mitosis. Now let's do a quick review of mitosis. If you remember mitosis from Bio-1, you might remember PMAT, prophase, metaphase, anaphase, and telophase. But it turns out that there's actually another phase in here called prometaphase, and that this is part of the bigger cell cycle that includes interphase and cytokinesis. Mitosis is the division of the nucleus, and we also call this like eukaryotic kinesis, division of the nucleus. So these phases are how we're going to divide the nucleus or the chromosomes. But before we can do that, we have interphase, which comes in 3 phases. And after we've divided the nucleus, we still have cytokinesis to divide the rest of the cell. So we see that this begins to be broken down into pieces. And a big part of what we're going to see is really high amounts of regulation to ensure that this is happening accurately. One quick thing, remember that sometimes the cells will actually still be joined or touching. But one thing we should know is these cells are, or in theory are, identical to each other. There's one other quick thing that we want to talk about, which is the idea that interphase can be the normal life of the cell. Most cells, most functioning cells, are spending their time in interphase. So if we think, for example, of a skin cell, the skin cell is not maybe actively dividing, except maybe skin isn't a good example, because skin cells do actively divide, but not every minute. Skin cells need to actually be doing their job and dividing when needed. So, when we begin to look at the cell cycle, it turns out that we can actually go in and out of the cycle into a stage called G0. And G0 is part of interphase, but it's known as the normal life of the cell. That's when the cell is going to be making proteins and doing its job. And if it receives a signal, it can re-enter. So the signal re-enters the cell into the rest of the division. So we do want to know that not all cells can do this. Remember, some cells, like liver cells, can go in and out. But other cells, like nerve cells, they rarely can come back in. So it's going to be important that we begin thinking about not all cells are the same. They have the possibility of undergoing the entire cell cycle, but we'll have to look at the exact conditions.